Hello, my name is Shawn King, and I am an undergraduate researcher in the Department of Chemistry and Biochemistry at UT Arlington in Dr. Chowdhury's lab. Today I'm going to be presenting my research poster titled Proteomic Analysis of Macrophage Cells Exposed to Perfluorooctane Sulfonic Acid. Per and polyfluoroalkyl substances are widely distributed in consumer products that are used daily, including cookware, carpets, clothing, furniture, and firefighting foams. It has been shown that these compounds pose significant health risks in humans, specifically organ damage, weakened immune responses, reproductive issues, and various types of cancers. The USGS reported a study estimating that at least 45% of the tap water in the nation has one or more of the 14,000 plus types of PFAS. Previous research has shown that PFAS can be bioconcentrated through different food chains with substantial contamination in fish. Perfluorooctane sulfonic acid, a specific type of PFAS, has the highest detection rate and concentration levels in both the environment and body tissues. The unique structure of PFAS is considered amphiphilic, containing a long hydrophobic fluorocarbon chain and a hydrophilic sulfonic acid functional group. The fluorocarbon chain is lipophilic in nature, allowing for passive diffusion across cell membranes and subsequent accumulation in body tissues. The persistence of PFAS in the environment and body tissues can be explained by the strong carbon-fluorine bonds, which require significant amounts of energy to break. Previous research has indicated a relationship between PFAS exposure and inflammatory markers in macrophage cells. More specifically, PFAS has been shown to stimulate the release of pro-inflammatory cytokines and inflammasome activation. However, the mechanisms by which PFAS affects the immune system are not fully understood. In this preliminary study, we have designed an in vitro experiment that exposes raw 264.7 macrophage cells to variable PFAS concentrations, and then we analyzed the proteins using liquid chromatography, mass spectrometry. In the methodology section, I've included a figure of our experimental workflow. So for a cell culture and PFAS treatment, cells were incubated until they reached around 50% confluency. Old media was removed and replaced with a new PFAS mixture media, and cells resumed incubation. Images were taken immediately after PFAS exposure, and then after 24 hours, more images were also taken, and then protein extraction and cell lysis. Once proteins were extracted, they were quantified using a BCA assay, which gave us the concentration of the proteins in each treatment group. After that, proteins were precipitated and purified using methanol chloroform, and then they were digested using trypsin enzyme. Digested peptides were analyzed using liquid chromatography and mass spectrometry, and identified proteins were classified and characterized using Proteome Discoverer and Panther classification database. In the results section, I've included the images that were taken in Figure 3. The top row shows the images that were taken immediately after PFAS exposure at time 0, and then the bottom row was the images taken 24 hours after PFAS exposure and immediately before cell lysis. Figure 4 shows the number of peptides commonly and exclusively expressed in different treatments, and Figure 5 shows the number of protein-coding genes that were identified and characterized in each treatment group using the Panther classification system. In conclusion, the identified proteins were successfully characterized using the Proteome Discoverer and Panther classification softwares. Characterization showed protein-coding genes primarily involved in binding activity, catalytic activity, cellular processes, and metabolic processes. Analysis also revealed proteins that were exclusive to the PFAS treatment groups. Small areas of localized cellular death were visualized immediately after PFAS treatment, but cells resumed growth afterwards. In the future, we hope to conduct more experiments using additional treatment groups, and we hope to achieve a larger number of identified proteins. This would allow